The overall goal of this procedure is to track photo-switched cells in a living zebrafish embryo. This is accomplished by first embedding an embryo expressing a tissue-specific photoconvertible fluorescent protein in low melting temperature agarose. Next, the protein within the region of interest is completely photoconverted. Then, the embryo is carefully removed from agarose and maintained in egg water at 28.5 degrees Celsius until later stages. Finally, the photoconverted embryo is again embedded in low melting temperature agarose. Ultimately, results can be obtained that show that photoconverted cells can precisely be detected at later stages due to the stable presence of the photoconverted protein through confocal microscopy.
So we think that the method that we will present today is going to be quite useful in the context of cell and developmental biology. So we think that using this methodology in the context of cell migration, tissue dynamics, and other cell behaviors will be quite useful. Demonstrating the procedure will be Verónica Lombardo, a postdoc from my lab.
After crossing transgenic zebrafish expressing a photoconvertible fluorescent protein, choose the brightest embryos, which are homozygous, and grow them at 28.5 degrees Celsius. When they've reached the desired stage, use forceps to dechorionate them in a glass container with egg water. To anesthetize